You know, for the longest time, finding new medicines was all about luck. But that's changing. We're in the middle of this incredible revolution where scientists aren't just finding cures anymore, they're designing them. I mean, literally building them atom by atom right on a computer.
Think about it. For centuries, drug discovery was pretty much a guessing game, right? Like searching for a single special needle in a massive, massive haystack. But what if you didn't have to search? What if you can just build a super powerful magnet that goes zing and pulls that exact needle right out? Well, that's pretty much the idea we're diving into today.
So, to really get why this new approach is such a game changer, we kind of have to look at how things used to be done. And honestly, the old way was a gamble, a huge multi-billion-dollar gamble that could take more than a decade to pay off, if it ever did. The traditional method is something called high-throughput screening. And really, it's a brute force attack. You take this giant library, maybe thousands, even millions of different chemical compounds, and you physically test every single one in the lab, just hoping something sticks. As you can imagine, it's slow, it's exhausting, and yeah, a lot of it is just waste. And this really shows you the difference. It's night and day. We're going from a game of pure luck, testing millions of things, to a game of skill and strategy. With computer-aided design, we can focus our efforts. We digitally design and then test a much, much smaller group of the most promising molecules. And the result is, well, it's everything you'd want. It's faster, it's way cheaper, and it is so, so much more precise.
Okay, so this all sounds great in theory, but let's bring it down to earth. Let's make it real. How does all this tech actually help with a disease that so many of us are worried about? Let's talk about the fight against Alzheimer's. So here's the thing. A lot of really complex diseases, like Alzheimer's, can often be boiled down to a single problem. One little molecule in our body, usually a protein, that's just gone rogue. It's not doing its job right. And the thinking is, if we can just fix that one protein, we might be able to stop the whole disease in its tracks. That malfunctioning protein, that's our target.
And in the case of Alzheimer's, one of the main targets is this enzyme called acetylcholinesterase. Let's just call it ACHE. You can think of it like the cleanup crew for the signals flying around in our brain. But in Alzheimer's, that cleanup process gets all messed up. So a huge goal for researchers is to design a drug that can go in and interact with ACHE in just the right way.
And this is where the precision we were talking about really comes into play. See, we don't have to affect the entire giant protein. We just have to hit one very specific spot, this little groove on its surface called the binding pocket. That's it. That's the bullseye. It's the exact place a drug needs to fit perfectly to do its job. Okay, so we've got our target, the binding pocket. How in the world do we design a molecule that can hit it? Well, the core idea is actually a classic. It's been around for over 100 years, but the way we're using it now is totally futuristic. It all comes down to a simple analogy, a lock and a key. And this, right here, is the star of the show, molecular docking. It's basically a super powerful computer simulation that plays molecular matchmaker. It takes our potential drug, that's the key, and it figures out exactly how it will fit, twist, and bind inside the protein's binding pocket, the lock. And that lock and key idea is just perfect, isn't it? Because for a key to work, it's not enough for it to be roughly the right shape. Every ridge, every groove has to line up perfectly. Well, it's the exact same thing in molecular chemistry. A drug needs the perfect 3D shape and the right chemical mojo to create that strong, stable connection with its target. All right, so if we're going to digitally design the perfect key, we first need a super detailed 3D blueprint of the lock, right? Makes sense. But where do scientists get these blueprints? I mean, we're talking about hundreds of thousands of different proteins. The answer is amazing. It's called the Protein Data Bank, or PDB for short. And honestly, this is one of the most incredible resources in all of science. It's a totally free, open to everyone, global digital library that holds the precise 3D blueprints for, well, pretty much every biological molecule scientists have ever mapped out. And check out the growth of this thing. It's just exponential. It took more than 10 years just to get the first 100 structures in there. But today, we're looking at over 240,000, this massive, constantly growing library of blueprints. This is the engine that powers modern drug design. So what does one of these blueprints even look like? Well, it actually starts out as just a boring-looking text file. Seriously, it's a long list of X, Y, and Z coordinates for every single atom. But then you feed that text file into a special piece of software and poof, it translates all that data into the beautiful, complex 3D structure you see here. And that is our digital lock, ready for us to start designing a key. Okay, so we've got our digital lock, the protein, and we've got a potential key, our drug molecule. So how does the docking software actually figure out if they're a good match? It really boils down to solving two big questions. First up is the stickiness question. Basically, how strong is that connection? The software calculates something called binding energy to score it. Then there's the fit question. A molecule isn't just a solid block. It can twist and turn. So out of all the millions of possible poses or conformations, what's the absolute best one? Now, to figure out that stickiness, the software just uses some fundamental physics. Think about two magnets. When they're a little bit apart, they pull together. But if you try to smash them right on top of each other, they push back. Atoms are the same. And the program is designed to find that perfect sweet spot, the exact distance where the attraction is maxed out and the whole thing is super stable.
And what about finding the best fit? This is where things get really, really cool. A lot of these programs use something called a genetic algorithm, and it's literally inspired by Darwin's theory of evolution. The software starts with a bunch of random positions for the drug molecule. It scores them all. The ones with the best scores, the fittest ones, get to reproduce and create the next generation of possible positions. You repeat that process thousands and thousands of times. And just like evolution, it naturally finds its way to the absolute best solution.
Okay, we've been deep in the weeds with all this amazing detail. So let's zoom back out to the big picture. What does all of this actually mean? What's the real-world impact for the future of medicine for you and me? Well, here's the bottom line. It matters because it's so much faster. We're talking about simulations that take a few hours on a computer instead of years of tedious lab work. It's way, way cheaper. You're cutting out all that expensive, wasteful screening of compounds that were never going to work anyway. And maybe the most important part, it's more precise. We can design drugs that are tailor-made to hit their target and only their target. And that means better medicines with fewer nasty side effects. And you know, what's really beautiful about this is how collaborative it is. This whole field is built on this shared knowledge. Every time a new protein structure gets added to that public database, every time we learn a little more about how these molecules talk to each other, it fuels the next big breakthrough. Our ability to design these new cures is completely linked to this incredible, growing, collective human effort. So that leaves us with a pretty mind-blowing thought, doesn't it? We have this global, digital library of life's most basic parts right at our fingertips. And the tools we have to work with them are getting smarter and faster every single day. So really, the big question isn't if we can design a cure for the next big disease, it's which one are we going to design next?